Hi everyone, welcome back. So we're discussing a very important clinical condition, which is glucose 6-phosphate dehydrogenase deficiency. It's very important. Before that, let's recap the pentose phosphate pathway, or also known as hexose monophosphate shunt. Now, it is divided in two parts. One is right here. This is the oxidative irreversible reactions, oxidative irreversible reactions. And here in the second part, you can see these are non-oxidative reversible reactions, right? That's what you can see. They all are reversible reactions. So in the first part, it consists of three important steps. So first, it goes like this, second and third. Here in this diagram, you can see they've accumulated the first two steps together and they've ignored one of the substrates. That's because that particular step didn't produce any NADPH, so they were like, let's ignore it because it's of no use for us. Anyways, so this is going to be an extensive, recap, and then we'll move to G6PD deficiency. So starting off, you need to understand that in HMP shunt, what happens is that we're using glucose 6-phosphate in order to produce ribose 5-phosphate and also fructose 6-phosphate and also glyceraldehyde 3-phosphate. Now, I'm sure you're very much familiar with fructose 6-phosphate and glyceraldehyde 3-phosphate because these are products of, not products exactly, but they're substrates of your glycolysis, right? So these will immediately or easily go and incorporate in the glycolytic pathway. And this ribose biphosphate, this is very important because this is useful for the nucleic acid biosynthesis. So it produces RNA, DNA, FAD, NAD, oxidized NAD, and a lot of other things. So this is important as well, ribose 5-phosphate. And another very important thing that HMP shunt produces is NADPH. Very important. So you need to know what all does it produce? It produces NADPH, ribose 5-phosphate, fructose 6-phosphate, and glyceraldehyde 3-phosphate. I hope that's clear. Now, when you talk about the oxidative steps, right here, which are irreversible. Starting off, you're going to have glucose 6-phosphate. Glucose 6-phosphate gets converted to 6-phosphogluconolactone. I know this name is kind of difficult and it's a mouthful, but just we get hold of it. So gluconolactone, right? So glucose 6-phosphate gets converted to phosphogluconolactone, and for that you need this very important enzyme that we'll be discussing, G6PD, which is glucose 6-phosphate dehydrogenase enzyme. Now, this is a very important step as well because this is the rate limiting step. Now G6PD, it is inhibited by NADPH. So if NADPH is high, so this is basically feedback inhibition because this step also produces NADPH. If you notice, the first step it also produces NADPH and also needs water. So it also produces NADPH, but the same enzyme is inhibited by NADPH. G6PD. So for that, this means this is product inhibition or feedback inhibition. Then phosphogluconolactone gets converted to phosphogluconate. It gets converted to phosphogluconate, and for that you need the enzyme hydrolase. So phosphogluconolactone hydrolase, right? Then phosphogluconate gets converted to ribulose 5-phosphate. It's ribulose, beware, it's not ribose 5-phosphate yet. So ribulose 5-phosphate. For that you need 6-phosphogluconate dehydrogenase. So what's the enzyme? Dehydrogenase. I hope that's clear. So initially also we had dehydrogenase and now again we have the dehydrogenase. So I hope these enzymes are clear. Now we got ribulose 5-phosphate, and then in the last step also the third step will again produce NADPH. So in the oxidative part or the irreversible part, we produce two NADPH, and we also produce one carbon dioxide, and we produce ribulose 5-phosphate. I hope that's clear. Moving forward in the reversible non-oxidative reactions, it gets converted to ribose 5-phosphate, and then you can also make fructose 6-phosphate, glyceraldehyde 3-phosphate, and you have transaldolase. This enzyme, it is capable of incorporating 3 carbons, and transketolase is capable of incorporating 2 carbons. So aldolase 3 carbon, ketolase 2 carbon. So that's it. There's nothing else you need to know in this part. That's the products. And now you may be wondering that why are we needing NADPH? What is its importance? So before moving to that, let's talk about, this is the pentose phosphate pathway. These were the two steps where we produced NADPH. This occurs in cytosols, this entire pathway, and it's ribose is important for RNA and DNA synthesis, and this occurs primarily in liver, adipose tissue, adrenal cortex, and gonads. These are the tissues which are active in reductive biosynthesis, and also it occurs in erythrocytes very importantly. Now, NADPH, how is it produced? So it's very important. It's produced by HMP shunt. The other way is by the help of NADP dependent malate dehydrogenase. So that's how NADPH is used. But why is it so important? It's important because It is needed for fatty acid synthesis, cholesterol synthesis, steroid synthesis. Then it is needed for drug metabolism, for the detoxification of drugs and toxins. And glutathione reduction. It is very important because it keeps glutathione in the reduced state, and that is important in order to um, you know, reduce oxidative stress on the red blood cells. Then it also helps with the generation of superoxides, especially in phagocytes, with the help of NADPH oxidase. And these superoxides, they are microcidal, or they are microbial killing, right? So microcidal, you can say. And NADPH oxidase is very important enzyme because if there's any mutation in this enzyme, it will lead to chronic granulomatous disease. NADPH is capable of inhibiting G6PD. Yeah, so that's it. Now let's come to a very important. Also here, I want to tell you one thing, that if you can see NADP positive is needed and you get NADPH. If you can recall pyruvate and lactate reaction, there we needed NADH and we were getting NAD positive, right? So this is one thing that you need to know, and you should be able to relate things. This is NADH, not NADPH, and we were giving. So that's the reason we say that like pyruvate or anaerobic respiration, in that you do not produce any NADH, you just produce 2 ATP. That's it, no NADH, because here you need NADH. On contrary, now when you talk about G6PD deficiency, what happens in that? So basically, this is a um, very important enzyme, and if there is any mutation in this, it can lead to hemolysis, it can lead to hemolytic anemia, because red blood cells are damaged. And okay, just let's look at this. So if there's mutation in this gene for G6P, the enzyme activity decreases, there is decreased production of NADPH. So everything roots from this problem, that there is decreased NADPH, because of which there'll be reduced glutathione, and we need reduced glutathione because that is what reduced glutathione actually prevents any oxidative stress on RBCs, and it prevents any sort of ROS, which is reactive oxygen species, accumulating in RBCs, or in simple terms, oxidative stress. It reduces it greatly. So because of which reactive oxygen species will increase, there'll be damage to red blood cell membrane, there'll be hemolysis, and there'll be hemolytic anemia. That's it. Trust me, this is just enough for you to know. And it's so simple. G6PD, very simple. You have to and describe it in regards to glutathione, right? So let me just explain it to you in proper words so that you can sentence it out and frame it out. It's a deficiency. It's basically a hereditary condition that affects mostly males, and it is characterized by hemolytic anemia when affected individual is exposed to an oxidant stress. So it only results in hemolytic anemia when a person is repetitively exposed to oxidative stress or oxidant stress. Now what happens is that the anemia is caused by the inability of the red blood cells erythrocytes to detoxify the oxidizing agents. When G6PD is deficient, there's less NADPH, which means there's less reduced glutathione, and then and also there'll be increased H2O2, because H2O2 is not getting detoxified, and it will lead to oxidant stress. Now, also one more thing, this is most severe in erythrocytes where the pentose phosphate pathway provides the only means of generating NADPH. So why do you think this is just, even though pentose phosphate pathway happens everywhere, why is this particularly affecting RBCs? Because in RBCs, NADPH's only source is pentose phosphate pathway. So pentose phosphate pathway is very important regulating enzyme. If that is deficient, then NADPH won't be produced in RBCs, because in RBCs we have only one way in which we could produce NADPH. In other cells, you have another mechanism by which you could, recover the shortage of NADPH. And the other thing is that this occurs mostly. This affects the males, and also in like babies. Once they'll experience neonatal jaundice, especially 1 to 2 days after birth, or 1 to 4 days after birth, they'll start experiencing neonatal jaundice. And also the people who are suffering from this disease, their lifespan is it may be somewhat shortened. And also a very important fun fact is that this deficiency can make a person resistant to malaria. Now, you may be wondering how's that. This is a very fascinating thing, and I'll tell you how. So, basically, malaria is basically a parasite, right? So, once that parasite it enters the blood, it will go and it will basically cause oxidative stress, and because of oxidative stress there'll be positive feedback and there'll be increased hemolysis and RBCs will be disrupted. So RBCs will be like, they will be hemolytic. And what will happen? Malaria, it can only complete plasmodium malaria, they can only complete their lifespan within the RBC. So if there's no RBC, how will they complete their lifespan? So that person will also be resistant to malaria, which is caused by plasmodium falciparum. Yeah. So, infection of red blood cells by the parasite induces oxidant stress, resulting of lysis of the red blood cells and protecting the host from developing malaria. Now, moving forward, another thing is that deficiency of this enzyme, it impairs the process of detoxification of the radicals and peroxides which are formed within the cell. Because of which, what happens is that oxidation of those self-hydryl group leads to formation of there's some sort of self-hydryl groups. Their oxidation produces Heinz bodies. These Heinz bodies are basically uh, ox- denatured proteins that get attached to the cell membranes of the red blood cells. So I'll explain it again. So deficiency of G6PD, it impairs the process of detoxification of free radicals and peroxides from within the cell. Now, reduced glutathione helps to maintain the reduced states of sulfile self-hydride, right? So reduced glutathione, it was helping with maintaining proper amounts of self-hydride, reduced states of self-hydride. But because of this deficiency, this will also get oxidized, and once this gets oxidized, it forms denatured protein. Those denatured proteins are Heinz bodies. Those Heinz bodies will start attaching on red blood cells, and they will make them very much more rigid and less deformable, because of which they're removed from the circulation by spleen and liver. So yeah, and also, what are the other precipitating factors in G6PD deficiency? So there's some uh, inducing agents, stress-inducing agents. First of all, oxidant stress, oxidant drugs. There are some oxidant drugs like antibiotics, antimalarials, analgesics, antipyretics. So these cause oxidant stress and they can ultimately lead to hemolytic disease. So for that, it's very important to give the patients a drug list to tell them and make them aware of the drugs that cause oxidant stress and to away from and so that they can stay away from those drugs. So yeah, first thing first is oxidant drugs. The patients need to be aware of them. The second thing is favism. What is favism? Favism is basically a hemolytic effect of ingesting fava beans. So there are three things that the person need to avoid. One is the drugs, the other is fava beans. So because of ingesting fava beans, the person can have favism, which is hemolytic effect. He can get hemolytic effect. And the third thing is infections. Infections is also one of the very important precipitating factors of the hemolysis in person in people with the G6PD deficiency. So what happens is that because of the inflammatory response to any sort of infection, there'll be production of free radicals, and those free radicals in the macrophages, they're going to diffuse into red blood cells and they'll cause oxidative damage. And yeah, so that's about it. And there's 400 G6PD variants discovered in the world, and still there is cloning and sequencing of this gene. It's there's research in that field going on. So here, this is one summary. Let's look at it. X-linked recessive disorder due to deficiency of G6PD, rate limiting enzyme of the HMP shunt, decrease NADPH and oxidative. It leads to decrease NADPH and increase oxidative damage to RBCs. RBCs depend entirely on HMP shunt for NADPH, and decrease NADPH, decrease reduced glutathione. RBCs become vulnerable to oxidative stress and it leads to hemolysis. What are the triggers? What are the precipitating factors in G6PD deficiency? What does precipitating factors in G6PD deficiency mean? It means the trigger factors, right? So you have drugs, infections, and fava beans. So drugs, antimalarial, antibiotics, and you had anti analgesics, you had antipyretics, and then we have infections and fava beans, which lead to favism. And yeah, that's it.